Hey, welcome to video number 22, the third in the mollusk series, um, following on from the prosopranch split into the archaeogastropods. So archaeo being older, this is a type that you're not likely to see. This is a Barotrachus species, and most of all of the archaeogastropods tend to be grazers. They pretty much all are grazers. So this one's grazing on a sponge. You could sort of call it predation if it's eating another animal. But when you're grazing, that means the thing it's not really able to run away. This is what slit snails look like. You're unlikely to see these. They're not very common in New Zealand, but we do have a couple. And you'll see the operculums here, okay, the little trapdoors. But what you will be familiar with are things like the paua, these top snails or top shells that this one's obviously tropical, but they can be really beautiful. But like the green top shell and the like are archaeogastropods. What do I want you to know about archaeogastropods? Really, that they're grazers, number one, and then also this older feature. If you remember our limpet underside, the bipectinate gill that I was talking about, it's got a big surface area because it's a feather, and if you think of a bird feather, they kind of go like this, and then like kind of like that, and they've got these striations coming down these gill structures, right, on both sides. If you think of feather, so bipectinate means that it's got them on both sides. Okay, and the problem with these things is on the mollusks is the bipectinate ones. They're easily damaged by sediment, and later on we find these monopectinate ones that are where it's only got one side and it sort of faces up away from the substrate, which means here's the substrate, which on uh, other ones of the snails, which means that they don't have as much gas exchange, but they do avoid the gill being damaged by the substrate. So archaeogastropods, the old snails like paua and things like that, they are unable to live on sandy or muddy substances. So if you're ever catching paua and you bring some up and they're undersized, make sure that you release them into hard substrate because they do not do well on sandy substrate. They'll probably die. We'll move on to mesogastropods and neogastropods. All right, they're different from the archaeogastropods in a couple of ways, but not so different from each other that we're going to go into the differences between the two groups. So most of what we see that's left, anything that you see that's not a grazer on the rocks, is going to be a meso or neogastropod. And the two major evolutionary changes that allowed them to that make them a little bit different are a siphon and also the monopectinate that allows them to go on to the softest sediments, the monopectinate gills, like was explained in the last video. But the siphon is very interesting. Siphon, and why is it important? The siphon is a fold of the mantle that sticks out, and it can be elongated quite a long way. And often you'll see these things swinging back and forth. But what it does is it allows water to be sucked in through the folds and lots of sensory uh, tissue, like essentially smell, just like our nose. As we suck air in through our nose, these suck water in, and they can essentially smell their way around the environment. They can smell their way to prey or to scavenge. You can see the muscular foot. There's actually an eye stalk, but the mantle is folded in. Here's the siphon that comes out. So how does that manifests into shells where you can see this long siphonal notch? This is a tropical murex that allows for a lot of sensory tissue. Here is a pinion whelk, very common in New Zealand. And by having these long siphons, they can sense their way to the prey. And when you see something like a cat's eye, here you go, the operculum, the little trapdoor that covers up the body and protects it. When you see a cat's eye or any shell that has a round aperture and doesn't have a siphonal notch, you'll know that's a grazer, and these ones are always scavengers or predators. So you can tell the difference between a scavenger predator versus a grazer just by looking at whether the shell aperture is round or has siphon. So we're going to move on to the gastropods that don't have shells now, and we'll start with the opistobranchs. Opisto means behind, and branch is gill, and that comes from the fact that they often have a gill tuft at the back part of the body, nudibranchs, one of the things that makes them look very pretty. Um, so bubble snails, sea hares, nudibranchs, sea slugs, and in some of them there's a shell, but the shell is internal. Okay, and some of them they don't have a shell. They have a gill tuft, but they also have tentacles called rhinophores. We'll look at those in a little more detail later on. Tend to be grazers, feed on sponges, hydroids, and other invertebrates. Okay, so bubble snails, common but often not seen. They have a little internal shell, and they tend to be small and clear, so often not seen. And then we move on to the sea hares, and just like they're really interesting. They're supposedly common, but I haven't seen a lot of them. It may be that they're more common on flat ground, and that is kind of where I've seen where we tend to spend our time diving on reefs. But this I've seen underneath mussel farms, and you'll see just like a squid, these things can release sort of a noxious ink as a way to avoid predation. But no, they're often called sea hares because they've got these two sort of bunny ears, antenna, and they are shellless. And then we get into the nudibranchs, where you see the gill tuft and the rhinophores, and these things are incredibly diverse and really beautiful animals. So they distinguish themselves, and one of their types of predation avoidance is just by being brightly colorful, like we've seen before. And what have we seen before when things are brightly colorful? It means that they're dangerous or poisonous. And these things often are poisonous as a way of deterring predators. Anyway, rhinophores. So rhinophores are the big antenna, the projections that come out at the front of the nudibranch. And when we look at the micrographs and look at them closely, we can see a terrestrial analog in a luna moth that's here and here. And you can see that they look very similar. So why are the terrestrial, when you see something that looks similar, it usually has a similar function. If you know anything about luna moths, they are essentially a phase of the moth that is only designed to find a mate, lay eggs, and then they die. They never even feed. They don't even have mouth parts. And they can find their way to a mate by smelling for pheromones using these antennas. And they can smell a female that's putting out some pheromones by as much as a mile away. So that's a kilometer and a little bit more than a half away. So you can see that the rhinophores, with all of this greatly expanded surface area, obviously have a sensory function, but they are essentially like antennas on the front of a nudibranch. So nudi means naked, and branchia, gill, again, naked gills, no shell, no mantle cavity, or the original gill is gone. And sometimes they have these projections called serrata, and the serrata increase surface area for gas exchange, but also can be defensive by containing nematocysts. So here is what happens if you've got serrata, then they use these as like a gill with extra gas exchange, and you won't see the gills on the ones um, that have serrata. Now, the amazing thing about nudibranchs, when we say they have nematocysts as a defense, is that they graze on sponges, which are nasty, toxic things, and they also graze on hydrozoans, which are cnidarians, and we know all about cnidocytes and nematocysts now. And it's incredibly, they can either package up the poisons from the sponges that they eat, not be affected by them, and yet use those as a deterrent. Like they package them up the poisons, and those become a deterrent because the nudibranch will become poisonous itself. Or they somehow can graze on the cnidarian tissue, the tentacles, suppress the cnidocytes from firing chemically, eat them, have them migrate out of their intestinal system through their tissue, and then be lined up on the edges of these serrata, ready to sting something that would attack the nudibranch. How they do that is like being studied, but it's just an incredible pathway of chemical suppression and movement to be able to move these cells all the way through their body of what they're eating as the defensive mechanism of what they're eating, and use it themselves as a defensive mechanism. Really incredible. And the nudibranchs, sea hares, they often leave evidence of themselves in these absolutely beautiful egg ribbons, which look kind of roseate. They look like roses or strips. And their defense is that they're incredibly noxious, full of noxious chemicals as well. But they stand out brightly. And you'll come across these while diving and wonder what they are unless you've seen this video, where if you look very closely, you'll be able to see the individual eggs as well when you do that. So now we're up to almost 13 minutes, so that's enough for this video, and we'll catch you in the next one.